welcome. We have Dr. Mike Kuiper from CSIRO Data 61 in Australia, as well as Daniel, our application scientist and structural biologist based in New York. And of course, I'm down here in San Diego. And we have the spike protein of the Omicron variant of the coronavirus that Dr. Kuiper has been analyzing. Every single mutation, the 38 mutations that have occurred in the Omicron variant, he's been analyzing them and trying to understand how they might relate to infectivity, immune system evasion, and all sorts of different things that could occur with all these mutations. But yeah, thanks so much, Mike. And feel free to keep explaining what you were explaining about that.
Thanks, Steve. It's wonderful to be here again. So as we were discussing before, this is my latest model of the Omicron variant, which I've been simulating a little bit. And what we have here is a truncated version of the spike. We don't typically do the membrane portion because that just makes the simulation too large. When we run the simulation, we also include all the glycosylations, and it's fully solvated. The whole idea of this is to try and understand how the various components, the various mutations are affecting the infectivity and the immune evasion, as you mentioned. So the big surprise, I think, with the Omicron variant is just how many mutations we see in the spike. So previous variants, like the Alpha and the Delta, only had about 10 or 12 mutations in the spike, where this has something like 34 to 38 mutations in the spike. So it's quite a lot more. And of course, when we have a lot more mutations, there's a higher chance of immune evasion. So by changing the surface look of the protein, prior exposure to vaccines or COVID infection means that you probably won't build a robust immune evasion immunity to the latest variant. Just like the flu. If you think about the flu, it often mutates from year to year. So this is sort of what we're seeing with the Omicron. It's just such a different mutation set. A lot of these mutations occur in the receptor binding domain. This is the cyan part here. The yellow part here is actually the ACE2 receptor, the human receptor that it actually binds to. Some of the new mutations in Omicron is in the arginine 493 and 498, as well as a nice one here as tyrosine 501 and histidine 505. I'll talk about those in detail. So I think the one that actually stands out here in terms of the receptor binding domain is the 493 arginine. Arginine being a basic molecule, it has a very strong propensity to interact with the glutamic acid and aspartic acid at positions 35 and 38 respectively. And this forms a very strong Sol bridge interaction, which we see here.
I feel like we talked about the potential when we originally analyzed this in like 2020 of new mutations forming, what might actually occur that would relate to the receptor binding domain. And now we're just really seeing this unfold over the course of the past 2 years. And now this is the big one. It's got all the mutations. It's got the receptor binding domain. It's got those cleavage area mutations as well. Like it's just it's got all the mutations that we've been looking for even a year ago, right? When we did the visit of all the variants, and we talked about the map of all the potential variants as well. I'm curious, have you done the comparison of the mutations that have occurred versus that study that had all the potential mutations that could occur?
Oh, with the STAR mutation, with the STAR paper, yes. So the STAR paper actually did point out, for instance, this 501 as a potential mutation, which would be quite advantageous for that receptor binding domain. And that's actually what we see in a lot of the latest variants. So that started with alpha. I think alpha had the 501 as well as beta and gamma. And yeah, we certainly now see it in Omicron. And in some ways, I think it's just the selection pressure that we see with the virus. We have so many people who are infected now.
Yeah.
And people with different states of health. So part of the thought was that someone who was immunocompromised had an infection which they could never quite clear. And then as the immune system is trying to take down the virus, it keeps replicating, and we get a selection pressure for a lot of changes. Then eventually the virus can break out. It's almost like when you take antibiotics to clear a microbial infection, but don't take the full dose. You just give it a half-assed dose. It just doesn't quite clear the system. So this is really quite a remarkable change, I think. And as we were alluding to before, there's a lot of changes that we have to sort of consider in these sort of models. So this spike protein has a lot of conformational flexibility. So there is this receptor binding domain in this region here. And that has to be in an up conformation in order for it to bind to the ACE2 receptor here. However, the spike protein can also be in a down conformation. So the spike is a trimer. So it has 3 of these receptor binding domains. And these receptor binding domains can be either in an up conformation or a down conformation, or any combination there or in between. So in this particular model, we have 1 in the up and 2 in the down. And when I look at the new mutations, you have to look at all the mutations in context of both the up conformation and the down conformation of, and put those mutations in context of that structure. So I like to think of the structure when it's in a down conformation. It's almost like a hunkered down, trying to evade the immune system, and maybe it's much better at evading the immune system, and maybe that's why it has this up-down conformation at all.
It's an incognito mode.
Yes, yeah, I think that's what it is, because when it is down, you can certainly see a lot more of this glycosylation covering the surface, which are hiding the epitopes. And also, a lot of the mutations are actually on the surface, on the up surface, when it is in the down conformation, providing even more immune evasion. So the ones that I want to bring your attention to is this 505H and a bunch of mutations here, which is occurring at 371, 373, and 375.
Oh, perfect. Yeah.
Yeah. So these 3 here are all occurring in this region. And when you look at that, you think, well, that's an immune evasion, and sure, it could be that. But when you look at it in context over here, where you've got a receptor binding domain in the down conformation, which butts up next to this N-terminal domain of the adjacent, you can see that actually this new cluster of mutations here at 371, 373, and 375 have created a new sort of little epitope here, have created a very ordered structure here. So where this was a bit looser before, it has now created a much tighter little motif, if you like. And this phenylalanine, for instance, is close to this tryptophan, so it creates a nice little pi stacking here. But interestingly here, you've also got this 505Y, 505H, sorry, histidine in this position here. And as we know, histidine is ionizable. So depending on the pH, it will be either ionized or not. And when it's ionized, it's more soluble. So at a more acidic pH, you would expect this to be a little bit more solvated in this region here because of the ionized potential. Which, if you look at this cluster of amino acids, which are all mutated, all the ones that are drawn as a ball and stick model mutations in the Omicron, we could potentially envision that this would be then solvated in a slightly acidic environment, which may allow the transition between up and down states. So it may be a new selective pressure here where potentially what's happening is when the virus finds itself in an acidic medium, which is often what we see in the nasal passages tend to be slightly more acidic than the lungs. Then maybe this can transfer into an up position. And if that's in an up position, then it can infect the cells. So this is speculative, but this is why we do modeling, because then we can ask those questions and then look for the experimental results. So if there are people out there who want to have a look at the...
Oh, sorry. Yeah. If that is the case, where a lower pH, more acidic environment would ionize it and sort of help it out and be more infectious, would you be able to take a prophylactic thing that would make your body more basic in certain areas,
well,
like a
hydroxychloroquine actually changes the pH, right? So I'm not supporting that drug, but I'm just saying.
Potentially. There's certainly there was a paper that I found the other day which was talking about a more alkaline nose buffering solution where you could do a nasal flush, and they were talking for Delta in that case. So I haven't read into it too deeply, but they had certainly suggested that. I think one of the things to try first is, of course, if you're doing a cryo-EM on these structures, people often look at the populations of the up-down conformations when they do cryo-EM, and perhaps if that was collected under different pH conditions, then perhaps we could figure out just how the pH affects the up-down conformation of that. And if that's the case, then that could be a potential treatment, is where maybe a slightly alkaline wash could help dislodge the virus from nasal passages or make it less likely that it can infect. But, that's something you'd have to look into carefully, because there may be other effects of... There's probably a reason why the nasal passages are slightly more acidic. So maybe you're opening yourself up to more different infections. So it's always a subtle thing there. So, but certainly these are the sorts of things that we start to explore once we look at the models. We can start to understand how the different mutations are occurring close to each other, and how they may interact with one another.
So, Mike, another interesting feature of these Omicron mutations, besides the mutations that you discussed over here, which is the interface between the RBD and ACE2, there's also quite a lot of mutations on the side of the RBD, which apparently have implications for immune evasion as well, right? Immune escape.
Yeah, that's true. Thoughts about that? So I guess it's when something's evolving, it's always a competition between retaining the infectivity but also evading the immunity. So it's trying to get the best combination of all of those things. And certainly there are a lot of mutations here which are exposed, which when we saw the mutations right away, we immediately thought this is going to be a problem for vaccines and prior exposure, because it's not going to give you the protection that you were hoping for. It's still going to give you some protection, but perhaps not as much as you like. And this is, of course, what we've seen with Omicron. People who have prior vaccinations and who have had prior infections are still getting infected by Omicron. Fortunately, they're not getting as sick as before, so that's a good thing, but they still are getting infected. But we're yet to see the people who have not been vaccinated. I think that would be a different story. If you haven't had any prior infection, I think this could be quite a serious infection still. It shouldn't be taken lightly. This is still a serious infection. And the other problem that we're seeing now, of course, is because it's so transmissible, and because it's evading the immunity, a lot of people are getting sick at once or in a very short time period, which is, causing a lot of problems at the hospitals. So we still don't want to, we don't want to see that volume of people getting sick at once.
Yeah, no, this is pretty much like a pandemic within a pandemic. Case rate in the US right now, as we're recording this, over 500,000 on its way to 1 million, it seems. Absolutely insane, just how infectious this thing is. And a lot of the people in the US, we're vaccinated. Like, I'm double vaccinated. I still got Omicron. It was milder because I'm vaccinated. I'm pretty happy I got vaccinated. But this thing is super infectious, and it's just ravaging communities, especially the unvaccinated communities.
Yeah, and as Dr. Fauci pointed out, even though it's less deadly, just by the sheer volume of people that are getting infected, even if 10% of the people that before need to go to hospital, because you're having 100 times more people infected, still like a 10 times surge on the hospitalization rate. So that's devastating. And everything else it's affecting, of course, and we're seeing that in Australia now too. A lot of elective surgery has stopped because they need to focus on the Omicron. Yeah, but there's certainly a lot of immune evasion going on here. And it's quite dramatic, I think, with the amount of mutations that we can see.
How many hours do you think you've spent looking at the various spikes in Omicron and Delta and all these other variants in VR?
VR. I often come into VR to really visualize it because, as you know, I'm a big fan of this technology because you can get a really good feel of this. And the funny thing was, when we started working on Omicron, we had the genetic data on the Thursday. On the Friday, I had started building the model. By the weekend, I had finished the model. By Sunday, I had gone into virtual reality with my friend Jason Roberts from VIDRAL, Victorian Infectious Disease Reference Laboratory. He's also a VR nut. And immediately we saw this sort of confirmation here, and we started speculating about what was going on. By Monday, I had the report for our guys at work.
Oh, wow.
So this was just such a rapid tool to visualize and to sort of make a preliminary assessment on how Omicron was. So within a few days, we had a report to my people to say, well, there's a lot of changes. It's probably going to be a problem with the immunity. There's some really interesting features going on here. And this was very quick. We should bring in Jason Roberts sometime.
Yeah, we definitely
it was
should.
Really
It'd be
fast.
Great to have Jason.
Yeah.
It was, and so I guess a lot of people are talking about like the data needs to be rapidly shared. We need to be sequencing and determining the structure, and giving out the sequence data so they could build their own homology models. Like, we need to do that rapidly. But then the other half of the equation is like, well, how do you actionize that data? How do you understand that data rapidly? How do you share that data rapidly? How do you build a report on if this is going to be a problem for Australia or not? So that's great to hear that you've been getting that rapid turnaround of valuable info into actionable results by using VR.
Oh, it was fabulous. It was so fun to just quickly jump in and immediately start to interrogate the structure, and with all the mutations mapped in, and we were very quick to be able to make predictions on some of the structural elements.
Yeah, cool. All right. Well, thanks, Mike. Really great seeing all of the interesting science going on with the coronavirus and the Omicron variant right here, getting hands-on and actually being able to see why it's more infectious or why it's evading the immune system. And this has been just really great taking us through this. And happy to do another one of these as the situation continues to evolve, and get more of your collaborators online, like you said. That'd be great.
Yeah, absolutely. I think with Jason, that would be very valuable as well. So certainly he's someone I work closely with, and we often talk about this in virtual reality. So it's just been a fabulous tool and very important for a rapid response and evaluation of these variants. I was going to say, see you next variant, but I don't know if that's a good
see
thing
you
to
next
say.
Variant. Thanks Daniel for joining as well, and to everyone at home, stay safe, stay healthy, get vaccinated, get boosted, and if you feel sick, try not to go out and spread it, and just stay in and try to just stop the spread. Thanks so much, and we'll see you next variant, unfortunately.
Absolutely. See you then.